Welcome to our video on digestion. So up to this point, we have talked about the structure-function relationship of biological molecules. We've talked about the membranes, how molecules and signals move across those membranes, and we're almost ready to talk about metabolism, which is that coordinated set of chemical reactions that serve to keep organisms alive by harvesting energy and synthesizing needed biomolecules. But first, we need to talk about digestion. And this is a set of biochemical reactions by which food is converted to molecules that then we can further manipulate to provide that useful energy or those building blocks. So digestion is the first step to get us to the metabolites we need for the metabolism cycle. So our goals in this video are to introduce the concept of digestion in the context of converting polymers back into their monomers, and outline the transfer of those monomer units into the bloodstream so that they can move throughout the body. So digestion is going to prepare these large biomolecules. So remember, as we think about those big biopolymers, we had lipids, polysaccharides, and proteins, okay, that we are going to be talking about. And we're going to break these down into their monomers. So fatty acids and glycerol, glucose and other sugars, and amino acids. Okay, that's the piece we're going to talk about in this video for digestion. After that, we're going to see, as we talk about our metabolism, we're going to see it gets converted into something called acetyl-CoA, which then get converted down here or get moved into the citric acid cycle. We make carbon dioxide. We create some ATP, some usable energy. We're going to talk about all of that piece in detail. But first, we got to talk about digestion. All right, so a little bit of review from a biology class. So your food is entering the stomach. As we think about the chemistry here, we know the stomach is really acidic. It has a lot of hydronium ion, H3O+. And now think about what we know about proteins. We said that large changes in acidity and pH denature those proteins. So it disrupts that tertiary structure and quaternary structure, if it's there, of those proteins. And when that happens and these things unfurl from these globular groupings, those amide bonds are more exposed to the enzymes, right? And let's go back up, right up here, and talk about just a couple things that I missed. And one of them is those enzymes, right? We know that for lipids, lipases break up those lipids. For polysaccharides at least, we'd have amylases. Okay, if it was like a disaccharide, it might be sucrase. Proteins, we have those proteases. So these enzymes are going to be key to breaking these things apart. All right, so then going back down and thinking in particular about those proteins, if we denature those proteins, those proteases will be able to better break those amide bonds. All right, so the stuff moves through into our small intestine, and we have various hormones that release digestive agents. So for example, we get bile from the gallbladder. We get proteases from the pancreas that help this digestion occur, and most of that occurs in the small intestine. If we look at these enzymes, right, we have some specific names of enzymes. We have where they're synthesized in the body, and this is just interesting because it's a term we've used before, right? Zymogen. So remember that zymogen is the inactive form. It allows your body to make a bunch of this inactive form of the enzyme and store it in various places in your body, and then when it's needed, it gets converted to that active form of the enzyme. All right, we're going to look at proteins first. So proteins are going to be acted upon by these enzymes, and they will get broken down either fully into those amino acids or into these oligopeptides. The amino acids can be ported into the cell right here. Okay. The larger pieces need to be further acted on, and they get broken down into either the amino acids or di or tripeptides and get ported into, and this is an intestinal cell. Okay, so we import it into those cells. And then once these things are broken down into those amino acids, we have an antiport mechanism that moves them out into the bloodstream so that they can be carried to various places in the body. If we look at saccharides, once those polysaccharides are broken down into monosaccharides, there's various porters that will pull these units into the cell, okay, so these glucose transporters, and then they are also going to get ported out of the cell into the bloodstream to get transferred around the body. Pretty straightforward. Clearly, there's a ton more details than that, but that's our basic idea. Lipids are a little more complicated, and it's because of their hydrophobicity, right? These things will form droplets in the stomach and the small intestine, and we need to be able to kind of break up those droplets so that the enzymes have access to the lipids to break them apart. One thing that helps us do is bile salt. So here's an example bile salt. And these things help break up those lipid droplets into kind of smaller groups. They give access to those lipases, okay, that will then help break them apart. If you look at this structure, hopefully you notice right here, ooh, it's that steroid. This is actually derived from cholesterol. Okay, so these bile salts are synthesized from cholesterol. So interestingly, one way to lower your cholesterol is to produce more bile and then drive it out of your body through a high fiber diet. Now, if you don't have a high fiber diet, your liver will reabsorb these bile salts and they'll be regenerated and reused. But anyway, that's a little aside. Let's talk about what happens to get to fully digest these lipids and get them into our bloodstream. So if we start with our triacylglycerol, so notice our glycerol backbone in black, and then we have 1, 2, 3 of those fatty acid chains. And a lipase in this first step will break off, what do I want? Right there. We'll break off one of these. So then we get to this diacylglycerol. We've broken off one of the fatty acids. So right here I have now a free fatty acid. We're going to see that happen again. And we're going to break off a second fatty acid. And then we're left with this monoacylglycerol. So we still have that glycerol backbone, and it has one fatty acid chain on it. Okay? And these things, remember, form micelles. So they spontaneously organize themselves to have the polar head groups outward and those nonpolar tails inward. Now, once that has happened, so we've broken up, here's the piece we just looked at. We've broken those triacylglycerols up into fatty acids and the monoacylglycerols. Those get ported into the cell. And then something called the fatty acid transport protein transports it into the smooth endoplasmic reticulum. And interestingly, what happens there is that these things get repackaged. They reform the triacylglycerols. So we couldn't get them into the cell that way, but now we're going to reform them and we're going to package them, okay, in these chylomicrons. And these are lipoproteins. a lipoprotein, so a mixture of lipids and proteins, and these get passed out of the cell into the lymph system and then eventually into the bloodstream, where then they get transported around mostly to fat cells and muscle cells. So this lipid process is more complex than the others. This is just all the words I just said. So maybe not in exactly the same words, but just so you have it there in writing. All right, that was our lesson on digestion. So next time now we get to jump into an overview of metabolism. Okay, and we're really going to be looking at this first part. We're going to look at glycolysis, where we start with glucose and get down here to pyruvate. So we're going to look at this little piece of metabolism to start. All right, see you in the next video.